And sort of a general comment about heterogeneity is, in the beginning, the dispersal, the net dispersal of these fish is primarily due to intrinsic randomness, but with time, the heterogeneity, the variance associated with heterogeneity absolutely swamps the variance associated with this intrinsic randomness. And that's an important concept. In the long term, heterogeneity in the populations, differences within the population will swamp out any internal randomnesses because it scales as time, whereas sort of Gaussian diffusion processes scale as a square root of time. So it's kind of an important lesson. So some penultimate quotes here. Incorporating heterogeneity is straightforward, yields better fits. It identifies some differences between species. There's some possible consequences for management. And population level heterogeneity dominates over diffusion at long time scales. So that was the fourth movement. At the end of some movements, there's this thing called a cadenza, where it's, what solos kind of riffs off on their own theme or something. So I'm going to do that for a minute in talking about heterogeneity. And that is to say that I've also looked at some data where they've looked at travel times and estimated survivals. This is kind of putting everything together. These are dams in the mid-Columbia River in Chelan County. Here's a dam, here's a dam, here's a dam. But they have detectors because they're not PID tagged, passive integrated transponder tags. These are acoustic tags, which are actually transmitting locations. And so they have detectors kind of in the middle of the reservoir here, right above the dam here, and a couple more sort of mid-reservoir locations. And just to give you an idea of what the environment is doing in a river, from April to August, the temperature is increasing, the flows are oscillating on a 7-day cycle because flow is tightly related to electricity demand in the Columbia River. There's some spill going through there, so the environment's sort of changing throughout the year. So there's kind of temporal heterogeneity in all of these aspects. And then travel times. So here's a dam, here's a dam, here's a dam, and we see that in different parts of the reservoir, so very crudely aggregated, in the tail race, immediately as it's released, speeds are much higher than here, where there's milling. Here we see that there's sort of clustering. So it's a group of fish spends 1 night, a group of fish spends 2 nights, a group of fish spends 3 nights. Right? There's a lot of structure and complexity within actual data if you look closely at it. I don't have a slide on survival. And the survival intensity, of course they don't have the slide here, is much different for different species in these different parts of the reservoir. And in a way this is kind of discouraging because it says, well, what's the use even of a model which is at a sort of scaled at a reservoir scale when there's all sorts of things going on in there. And I think it's just important to be aware that these environments are really heterogeneous, and to really pick out true processes going on inside, it's really worth investing in looking at different regions within a given reach, looking at different kinds of interactions and processes, looking at getting as much accessory data as possible. A lot of money is invested in the Columbia River on salmon, but it's remarkable how little you can learn about how the predators are distributed, for example, and how little is done on sort of scales of migration within a reservoir. There's certain things that are kind of done, and other things that aren't. So if I was going to sort of give a recommendation to managers, I would say throw a little bit more money in finding out and parsing out all the possible variables going on within the sort of migration survival process. So that's the end of the fourth movement, the last movement. Here's a coda. It's a nice coda. Don't consider that anything has been done if anything is left to be done. That's a little bit hardcore, but it's also, I think, relevant to me because at the end of the day, kind of ask myself at the end of the day, at the end of 6 years, what have I really learned about a system? I certainly can't explain red tides based on heterosignal movements, and the fact that fur seal behavior is complex is pretty self-evident to anybody that actually hangs out with fur seals. And there's differences between quantifiable differences between the migration behavior of steelhead and chinook. Is also kind of self-evident. People that hang out with steelhead and chinook. There's the reason, part of the reason that we're interested in animals and behaviors and movements is because they're complicated, because they're heterogeneous, and because there's a lot of variability, and they're not just particles bouncing around hitting off each other. And because of that, the appropriate ways to think about that, model this, and think about this sort of data, you really need sort of a mathematical approach, so you can have some sense of it. But because of all the messinesses involved with the real world, there's some sort of statistical sort of aspect to it. And integrating that all is sort of what our purpose is as quantitative ecologists. In this book-like thing that I am in the process of finishing up, I present some tools, some methods, some ideas, based on this idea that you should look at the organism sort of as its center, and you should think about, all the complexities and not run away from them, and you can potentially learn a lot. And I hope that when it's all kind of done and said and compiled, that there's going to be some applications that will have relevance. That's pretty much the best knowledge. So that's the Coda. Acknowledgements, yeah. So I want to thank the committee. The committee is really diverse group. I haven't had any interactions with any of them that haven't sort of gotten my mind racing. It's greatly appreciated, and Jim's been incredibly indulgent. I've gotten
big salary when you were in jail.
I've gotten data from a whole bunch of different people, lots of different sources, and that's been exciting to collaborate in different ways. I'm really looking forward to collaborating further with a lot of people, learning more about the active collaboration, because it's really important. Funding, a lot from the Monroe Administration, from Quorum Grad School, NIMS, and Marine Mammal Lab. Inspiration, from peers and friends at Quorum. Quorum has been a really great environment for learning, in particular because of the peer. The peer is a group of people that I've gotten to be around, interact with, just bottomless source of ideas, concepts, and practical advice and just support, and all over, in the School of Fisheries, in CFR, and biology, and more places. I've had really, really useful, positive conversations with a bunch of other miscellaneous adults, some of which are listed here. I'd like to thank my family, who unfortunately can't be here, for everything. And, the full moon. It's the full moon. The full moon is the only day in this entire summer that my entire committee could assemble. So, shout out to the full moon. And to the animals for bearing so game with all the poking about. That's it.